we're gonna have the next section starting up. The first talk is given by Francis Ding, who's now at Harvard, but starting at Berkeley soon.
Thank you so much. Thanks to the organizers for inviting me to talk. So today I am going to be talking about some recent work, so recent it's still in progress, called Representation via Representations. So this is joint work with Cynthia Dwork, as well as our collaborators at the Harvard School of Public Health, Giovanni Parmigiani and Prasad Patil, who are experts in biostatistics, particularly genome studies of cancer, and have really provided a lot of inspiration for the problem that we're addressing here. So I want to give a quick outline for what I'm going to talk about today. First, I want to motivate what is the question in biomedical prediction that we're trying to solve here. Second, what are the challenges in out-of-distribution generalization that we face? Then I'm going to take a detour into adversarial methods that have been developed in the fairness literature and describe how they can be applied to this problem, how we've incorporated them into our representation via representations model, and then some preliminary results we have, and then conclude and provide some future directions. So, what is biomedicine up to today? Well, over the past couple of years, there's been a lot of hope for precision medicine and personalized medicine, including the 2015 State of the Union address by then President Obama announced this huge initiative to provide healthcare tailored to you, to move away from this model of medical treatments designed for the average patient, but to really take into account individual differences and allow for medical treatment to be tailored to each person. And it's really big business as well. So the global cancer and tumor profiling market is projected to reach $12.4 billion in the next 5 years, from already billions of dollars right now. So there's a ton of hope. What has come from it? Well, there's a lot of worry still. So there's a worry about whether these biomedical predictors, which come out of academic studies, actually turn into real results in the clinic and real treatments for real people. So one thing that has been pointed out is that cross-study validation indicates that conventional methods of estimating how well you're going to do on future populations, conventional cross-validation, is overestimating accuracy. In case people aren't familiar with these two ideas, I'm going to go and describe both cross-validation and cross-study validation really quickly. So cross-validation is this idea that if you want to figure out which algorithm is going to work well on your problem, you want to choose between a logistic regression, a random forest, a neural network, you want to choose the right hyperparameters for that model, you're going to want to test on some held out data to make sure that you haven't overfit on your training data. So you might collect a data set and you might hold out some of it or train on some and then hold out others as test. But you don't want to just be biased towards this random 20% that you held out for a test. So maybe for cross-validation, you actually iteratively hold out a different segment of your data set as the test, and then you average the performance of your classifier on all of these different folds, and then you figure out, hey, logistic regression did the best, and now I'm going to retrain it on the entire training data set and use that in the future. So cross-study validation takes this one step further by saying you get inflated estimates if you just look at one data set that's been collected at one time from one hospital. What in fact is going to tell you a better measure of generalizability is if you collect many different independent data sets. So I'm trying to indicate that with different colors. So different hospitals, different people collected at different times. And at this point, you do a similar thing. You hold out different portions as the test set, and you calculate average performance, and now you want to see which is the algorithm that does the best, what are the hyperparameters that I should use. Unfortunately, the ranking of the best prediction algorithm often changes when you compare cross-study validation versus conventional cross-validation. Simple predictors often do better than complex ones, so your linear regression often does better than a neural network. Meaning that although you thought that you had properly defended against overfitting, you probably still are overfitting. And this is happening in the real world. So biologists have raised alarm. The biomarker discovery field is replete with molecular signatures that have not translated into the clinic. And this continues to be the case. So this was observed in 2014, but it's still happening to this day. And what are the fairness implications of this? Well, biomedical studies tend to be conducted in large research hospitals, not rural clinics. And precision medicine will only benefit these segments of society unless we explicitly try to improve the applicability of these predictors to unrepresented populations. And unrepresented is really important because there are some groups that are not just underrepresented, but really not in any training data set at all. One example of this is the Havasupai Indian population, who for historical reasons are not willing to participate in future biomedical studies and will not be present in any training data sets. But I would claim that it's a fairness concern if they're not going to have accurate predictions coming out of these personalized medicine initiatives. So why don't predictors generalize? I want to walk through a couple of the reasons, not a comprehensive list, but some of the reasons that biologists have highlighted to say these are some causes for why generalization is hard. So one thing that happens are these mysterious batch effects, which are just systematic differences between samples that are processed and measured in different batches. And it's kind of a vague term, but it just means even if you control everything you could think of, even if you grow a cell culture from the same line, have the same technician handle them, grow them for the same amount of time in the same lab, but just a couple hours apart, you will often see batch effects. You will often see slightly different gene expression values when you then do some kind of DNA assay on this later. Aside from those kind of batch effects, also there are things that make a lot of sense. If you use different equipment, if you use a different company's assay, you will often get different results. If different technicians handle samples, if you have biological variation in the underlying populations that creates spurious correlations, not really related to your phenotype, but it sort of messes up your classifier, or if you have also phenotype-associated biological variation, so things that are actually related to your outcome of interest. And finally, if you have really noisy measurements so that you have crappy data. So, still a problem. In the community, there's been a lot of growing interest to address this problem. Some proposed solutions include explicitly trying to train a simple classifier that's geared towards reducing the possibility of overfitting. So one example of this is this Maso-Menos method, where you calculate a risk score based on the sum of sort of bad features that seem correlated with a bad outcome, minus the sum of good features. And so instead of doing the linear regressions, it's almost like everything just has a coefficient of +1 or -1. Another example is training replicable predictors in multiple studies, where you train an ensemble of predictors, you explicitly have access to multiple studies, and then you use a cross-study validation kind of ensembling to make sure that you're weighting by the ability for a particular predictor to generalize out of sample. And finally, one more example is embedding independent validation data sets within a study. So explicitly talking about validation to out of sample rather than, and collecting this independent sample yourself and holding it out. Okay, so now I'm going to take a quick detour into talking about some literature on fair representation learning, which was covered a little bit by Rich Zemmel yesterday, and I'm going to tie this back later in the talk to see how it addresses the problem that I've outlined so far. So what is the goal of fair representation learning? We want to learn representations that censor sensitive information. So the idea is maybe we want to allow a system to use a classifier to classify people, but we're pretty afraid of some malicious actors. If they have access to this sensitive information, maybe they can discriminate on the basis of it. And so what we want to do is learn a mapping from data to representations that stamps out sensitive information, but at the same time retains other useful information for classification. And so we can hand this representation to anyone and be sure that they're not going to discriminate on the basis of these specific sensitive features that we've isolated, that we've censored out. So this may not be the right fairness notion for all situations. In particular, there are a lot of cases where you might want to be aware of sensitive attributes rather than censoring them out, but it is entering real-world use in a lot of cases. For example, San Francisco using NLP to automatically redact information from police reports that could identify a suspect's race. And that's some of Sharad Goel's work with the Stanford Computational Biology Lab, and about to be rolled out. Sorry?
Computational policy, not biology.
Oh, policy. My mistake.
It's your own bias.
I'm in the biology zone right now. So one example of this learning fair representations approach is using adversarial learning, so adversarial neural networks. In this setting, what we have is, imagine you have a data set X, and it's made up of green people and blue people. And green people and blue people also have a sort of shade to their color. And the shade is what we're going to say is relevant for their classification. But their color, green or blue, is maybe the sensitive attribute that we don't want to have left in the representation. So what we're looking for is a way to map from X to our representation Z through this encoder, some black box neural network, that is going to preserve the shading. We still have dark and light sections, but it no longer shows whether you're a green person or a blue person originally. And how we train this is we have a multi-objective thing going on. So we want this representation Z to be relevant to our outcome of interest, Y, whether that's providing a loan to someone or whether their tumor is going to metastasize. And so we are going to make sure that there's a predictor that does well predicting Y hat. At the same time, we want to make sure that Z is no longer showing the green or blueness. So we have an adversary who's trying to guess all the time whether someone is green or blue, which is A hat, the attribute. And then finally, we also have a decoder, which is trying to start from representation Z and go back to X and ensure that the representation Z is still holding on to as much information about X as possible.
What does it mean as much information as possible? I mean,
so,
You just go on, it'll be clear from the loss function.
right, so essentially you want reconstruction to be possible and you want to minimize reconstruction loss between the two. Okay, so the loss function eventually that you, at a high level that you're trying to optimize is some hyperparameter alpha times the loss for the predictor, maybe a 0-1 loss or a mean squared error loss, some beta term times the loss of the decoder, which is like a reconstruction loss, and then a term for your adversary. So that is a way that you can trade off these hyperparameters to see which thing you value more in a particular optimization. Okay, so that was learning fair representations. What does that have to do with the problem that I was introducing earlier? We have a hypothesis that for certain biomedical tasks, there exist predictive invariant features that are invariant across populations. And this hypothesis comes from not just thin air, but a lot of conversations with Giovanni and Prasad and other people in the biomedical space who really think that this is a reasonable hypothesis to claim about certain biomedical tasks, such as predicting tumor prognosis, predicting 5-year survival rate, certain things like that. Debulking, so that's about whether surgery is a good option to treat your cancer. And so using this hypothesis, we conjecture that techniques for censoring sensitive information in the fair representations literature can help us to learn predictors that will be accurate on unseen populations. And how we're going to do this is use data from multiple institutions or multiple studies done at different hospitals and use that to censor out study-specific features and build a predictor based on these invariant features. So this is starting to sound a little bit like domain adaptation, which some of you might be aware of. So I want to highlight some differences with a lot of that literature. Domain adaptation is about using data from one source distribution to transfer to a particular target distribution. So people are really interested in this because a lot of times we have, for example, ImageNet, a huge data source collected in the Western world, but we'd like to still be able to do image recognition on other places in the world, and so we want to use data from this source distribution to still be accurate later on a new target distribution. The common settings that we see domain adaptation at work are to claim that, okay, if we have abundant source data X as well as labels for this source data, and a small amount of target data and labels, then we can transfer with this setup. Or, secondly, we might have abundant source data and their labels, and abundant, or at least a significant amount of target data X, but no target labels Y. And what you're doing is hoping that if you make your source data X look like your target data X, then the labels will also transfer. But in our setting, we don't require any training data X or labels Y from the target population. What we're relying on is our hypothesis that there really are invariant features and an invariant function that goes from features to the label in our target population.
So this is the area which called transfer learning that's been studied a lot for like recommendation system. So I have lots of people that I want to create, I get data from, and then I want to recommend to other people, and I don't know who these other people are going to be, and I don't. So in the literature of transfer learning, this scenario has been studied.
So a lot of transfer learning tries to transfer still using knowledge of the future distribution or making assumptions on future
Also,
distribution.
just extracting invariants.
Right.
Good.
Yes,
That's
great.
Now that you say it, it's familiar. I didn't. So
I can give you a
tell me the best place to look for the current state of affairs.
Okay, sure.
Thank you very much.
I'm a little confused about how the existence of invariant features sits well with the goals of precision medicine, where the idea is that there's something particular about each individual that you're going to use.
So the goals of precision medicine often are saying that there are specific gene biomarkers or specific things which aren't necessarily present in everyone, but mean the same thing for across different people. So that if you were to present with this biomarker, it might only be present in a fraction of the population, and that's why it hasn't been studied at wide scale now, because it's maybe an orphan condition. It only affects a small proportion of people, only a small proportion of people have this biomarker, but when it's present, it really indicates something about disease. Yes?
So like, on its face, it seems like sort of what you'd really want to do is learn like a causal model that relates, individual people's features to whether they're going to develop some disease. And this actually sounds like it might do that. What have you thought about?
Yes.
Under which you might be able to prove that?
Right. I think it would be great to be able to get at a causal interpretation of this. We don't have any formal connection with it right now, but it's a question that we're interested in.
Francis, just a comment to that question. So there has been some recent work called invariant causal prediction, which essentially works under this assumption that you have some invariant features across different populations, and then there is a community of people who try to reason that they believe those are the causal predictors because it's kind of stable across populations. So I'd be happy to
Okay,
chat
that would be
on
great,
this
yes.
because I think there could be a good connection of this work to that, in which case we'll have a causal interpretation of it.
Great. Okay, thank you. Great. So quickly, a return to what I was talking about earlier about why predictors don't generalize and whether our method addresses these sort of issues. So we have batch effects still. Can we censor out these batch effects? We hypothesize that we can. Different equipment. Let's censor out equipment effects. Different technicians handling samples. Let's censor out these technician effects. Non-phenotype associated biological variation in the underlying populations. We should be able to censor these out. Now the story changes a little bit when we talk about phenotype associated biological variation. And the story is a little complicated because our hypothesis may or may not apply. If there's really biological variation that we have no data on and that's related to the outcome, it's possible we can't generalize well to them. And you would really need to validate a model on a new population to assess whether this is the case. Also, if you have noisy measurements, you need to get better data. So what is representation via representations trying to do? We have now an X that's made up of many different studies coming from many different hospitals, for example. And we still want to do a similar thing, learn a mapping to a representation Z that gets rid of that study identity now, is our sensitive attribute, but still retains the information relevant to classification. And then we train it in a similar setup with a predictor, adversary, and decoder. And at test time, what we do is we freeze the encoder and the predictor. Now we have a new study, study K+1, a new color, but we hope that this is still going to work, that it's still going to map down to a useful representation Z, and that it's still going to be an accurate prediction Y hat. And so the specific loss function that we use for the adversary is here tailored to a multi-study setting where you have sort of a study normalized loss where the adversary is trying to guess from the representation what the true attribute was, and going in reference to Madras et al. And the original learning fair representations work, this can directly connect to, this can provably connect to fairness notions, it can provably connect to learning invariances. And so A here is a one-hot encoding of the study identity. So for example, what that means is study 1 out of 4 would have A equal to 1, 0, 0, 0, and study 2 would have A equals to 0, 1, 0, 0. So in our setup, we're motivated by this biomedical setting. Working with real data in this setting, though, is often difficult because of the very small sample sizes. And so a lot of the work from our collaborators has been on synthetic or simulated data that comes from realistic data. And so we followed a lot in that mold to start out looking at synthetic data. And so now I'm going to describe how we generated that synthetic data and why it sort of makes sense in a biological context. So let's imagine we are now going to look at sort of one row out of our entire data set. So this is one hospital's input, or one input from one hospital. Blue is going to be study I. So this is coming from the blue hospital. And we're going to assume that the X features can be split up into invariant features and study-specific features. And that there's an F invariant that applies to the X invariant and a series of different F primes that apply to the study-specific features, K different ones for all the K different hospitals. To restrict our attention to our blue hospital, let's look at just F prime I. So how we generate the data is we choose a base rate for study I. We draw Y according to a Bernoulli, and we have a different base rate for every study, so that's one source of heterogeneity. Then we draw X from this normal distribution, also different for each study, and draw an epsilon noise term from a standard logistic distribution. And what we do is some rejection sampling. So we only retain this XY pair if the Y that we've drawn is equal to sort of the outcome of both the study-specific and the invariant function, where the study-specific is just left alone, but we have this noise term that we add to the invariant function, so we make the signal a little weaker in the invariant function. In this way, we're trying to make sure that our whole setup is actually learning the invariant function even when it's almost incentivized to learn the study-specific function. Any questions here?
What's the mapping of F?
Right, great question. That's the next slide. So what are these Fs? The ones relevant for genomic analysis seem to be largely based on linear functions with a few interaction terms or complications added on top. So most biologists think that a lot of phenotypes can be explained by largely linear effects. So what we looked at is a completely linear F. We also looked at linear with a few pairwise interaction terms, including product interaction terms. So gene 1 times gene 2, and logic interactions, so are both gene 1 and gene 2 above a certain threshold, and also composites of two linear functions. So if we take half of the invariant features, apply a function to them, apply another function to the other half, and then compose them with an or or an and.
When you say a gene is above a threshold, do you mean like the protein expressed by that gene
Sorry,
is above
yes.
a threshold? Okay.
Gene expression value, yes, exactly. So always the signal from the study's specific features is stronger than from the invariant features to try to really stress test our model. So what are some results? Starting out with completely linear functions, this graph is trying to show a 3D plot where we change the fairness coefficient. So if you remember the loss function from earlier, this is the coefficient that affects how much the adversary is trying to tamp out the study identity. And then the reconstruction coefficient is for the decoder to try to make sure that the representation Z is able to reconstruct X well. And so in this 3D plot, we have the representation via representations model here. We see that at fairness 0, when it's just a free neural network, it seems to overfit. It does pretty poorly on this totally held out test set, but as you increase the fairness coefficient, as you force it to censor out the study identity, you see that its performance gets better, its error decreases. And for baselines, we have a random forest and a logistic regression. These were trained on just the merged data sets, but we also have baselines where they're trained using a multi-study ensembling method that's trying to really explicitly take into account that they come from multiple studies, and they do pretty much the same. So to not clutter this graph, I didn't include all 4 lines.
So it seems from here, I mean, it's hard to see this 3D graph, but it seems from here that you just want to crank up that fairness coefficient as high as you can. And I mean, the best performance is obtained at that corner closest to us.
Mm-hmm.
Is that just because the 3D
Right.
plot
So
changed, or what's
yeah, I think it's hard to see. It actually does start to go up at the end. So there seems to be a sweet spot for the fairness coefficient. If you crank it up too high, performance degrades as well. And I'll show some more graphs which illustrate this a little bit better. And in this case, when it's a completely linear function, we see that logistic regression almost has an inherent advantage because it's the model that the data is really being generated from. So it has this inherent advantage. So moving on to slightly more complicated terms, we have a linear function with an interaction term. In this case, we actually see that the representation via representations approach always does better than our baseline, so maybe just the neural network is already great. When we look at a cross section, so I took one reconstruction coefficient value and just looked at as you vary fairness, we see that you start out doing pretty well, but you do even better by increasing your fairness coefficient. So a similar qualitative result as we saw in the previous slide. And we see that interaction terms are important for getting rid of the advantage of logistic regression. Now, one other example of a data set that we looked at is if we take 2 linear functions and take the logical OR of them. And I think this one shows the nicest curve of accuracy of error decreasing as you increase fairness coefficient, but then not changing very much. And you seem to do pretty well without having to crank it up to infinity.
Maybe I missed something, but is there a reason that you don't plot the RVR for the baseline models?
Sorry, you mean I don't have like a color graph?
Yeah.
So the baseline models don't have any hyperparameters that I tuned to show in this graph, so I just plot them at like a single hyperplane, if that makes sense.
Okay, maybe I'm confused about the graph.
So the coloring is just to help you see this surface a little more clearly. And there's no coloring for this one because it's just going to be a flat. Okay, so a complication though is that when we add many more studies, so all the previous graphs that I showed were dealing with 4 training studies and then 1 totally held out test study. And when you have many more studies added to the training. This was with 10 studies in training, one 11th study held out for test. What you start to see is that the random forest actually does pretty well, and RVR doesn't do terribly, but it's not that great. It's significantly worse than RVR, or than the random forest. And what we think is going on here is that the random forest has been pretty limited in its scope, and it can't overfit to all 10 different training studies, and so it starts to be forced to rely on the invariant features as well.
So are there scenarios where setting the reconstruction coefficient, say, large, actually improves things significantly? Because in a sense, if you knew what the task was up front, then I don't care about reconstruction, I just care about the end given by the representation.
Right. So in the linear case, the actual best point for RVR is for non-zero reconstruction. I don't know if that's like easily visible, but we do see it in some cases, and that's a really interesting question as to why reconstruction helps when we're not transferring to a different task, we're actually trying to do the same task. Okay. So, so we think the random forest has somewhat of a benefit when you add many, many more studies to your train set, and also we think that there are some difficulties with the adversary training when you have this many studies. Because of our data generation setup, all our x's come from Gaussian distributions with slightly different means, but we think after you add a certain number of them, it gets hard to really pin down exactly which Gaussian it came from. The adversary has a difficult time, and the adversary is no longer forcing the encoder to do a good job of censoring. So another example of that with 10 studies. So these were both cases where RVR seemed to do better than both baselines without very much hyperparameter tuning. And then now in the 10 study case, we have a lot more difficulty. Okay. So to conclude, main takeaways are there are many different reasons a model might fail in a new context. Some of these reasons are due to a failure to find the right invariant features. And adversarial methods developed in the fairness literature to learn fair representations can actually help learn these invariant features. However, I still think I would argue that models should be validated in new settings if possible. The representation via representation method, though, can create a better starting point, can make sure that you're overfitting on as little dataset specific noise as possible. And finally, I think there are a couple extensions of this potential work. So still in the biology space, you could imagine that X-ray images taken at different hospitals or taken on different equipment might show a similar structure of variation. The Gender Shades project to try to transfer image recognition and face recognition across different contexts, and any other settings where really you expect invariant features. And as others have mentioned in comments and questions, I think a causal interpretation would be really great. Okay, so thank you very much.
We have lots of time for questions.
Yeah, cool. So first, that was a great talk and really interesting. So I don't know much about the domain, but like I would guess, like in your example of urban hospitals versus rural hospitals, that there's lots of different demographic differences, like the age distribution in rural hospitals is probably different than the age distribution of people who show up at, race. And like I would also guess that these things are, at least age is sort of relevant to some of the precision medicine applications. And so like, you might worry that like if the sort of regularization you're proposing is to stop anyone from being able to like guess better than, like a randomly which study some data point comes from. They could get like some advantage just by, guessing that the study that you came from was the one in which your sort of demographic population is most frequent, and then stopping that kind of advantage seems like it would hinder you from being able to target features like that as part of the...
Right, so so the loss functions I showed were for that kind of demographic parity idea where you really don't want to do better than a coin flip, but the loss functions we use more often in those experiments are actually more akin to the equalized odds framework. So you want to make sure that of the positive classifications, you can't tell between one group and another. And so hopefully that gets around some of the issues of, really indicative features being censored out.
Okay, that was great. One quick, hopefully, clarification question in the comment. So you had, there's one thing I didn't understand. You had this slide where you mentioned all kinds of things you'd want to censor. Where do these come in? Because at the end, in the method, if I understood correctly, you're just basically, you have a bunch of different studies and you want to kind of just censor what the origin of the study was. So is that just, should I interpret it just as motivation, or is that actually goes into the methodology?
Right, it's just motivation and trying to sort of train intuition about where this variation is coming from and which settings we might actually hope for this method to do well versus fail.
So it's not that when you use it, you actually make this list of different technicians.
Right.
Now, so in the comment that relates, I think, also to what Shai mentioned, I think one possible baseline here would be think in terms of star-based multitask learning. So multitask learning, usually we use that to generalize when they have multitasking, when you transfer between them. But another way to think about that, if you think of each study as a different task, and if you're setting up the problem as a star-based multitask learning, meaning in star-based multitask learning, you roughly have one base predictor, and then one specialized predictor for each study, where you want to have the specialized predictor be similar to the baseline predictor, the specialized predictor be somewhat similar to the baseline predictor. If you try to fit a specialized predictor to each study and then use the centralized predictor in order to generalize to new studies, that also has the effect of removing the kind of study-specific stuff that would hopefully be captured by the variation between the specialized predictors and the centralized predictor. And so I'm wondering if that might be a good baseline also to compare to here, and might be a relevant approach. I mean, that is kind of a standard approach where you think you have data that's coming from a bunch of sources that have some commonalities, but also some differences between sources.
Right. That makes a lot of sense.
And there is standard methodology for that. So.
Does that use training data from the new distribution?
No, so you don't. So the way you use here, you have training data from. Think of each one of the studies as a separate task, and you have training data for each one of the studies, and you're going to learn a different predictor for each one of the studies, where you're also going to constrain all these different predictors to be similar to some centralized predictor you're also going to learn. So you don't need training data from future tasks. You just have training data from all the tasks.
Okay.
We can maybe
Makes sense.
go over the details later. I can give you some references.
All right, more questions? Thanks, Francis, again.